The chemistry of Pumplox virus. Pumplox virus is a member of 40 virus genes and has a positive sense single stranded RNA genome. The virus particle is filaments in shape and measure approximately 750 nanometer in length. Pomplox virus is transmitted by the aphids and can infect a wide range of plants species, including plums, peach, apricots, cherries. One inside the plant, plumpox virus replicates in the cytoplasm of the infected cell and causes the damage to the vascular system, resulting in the stunted growth, reduced fruit quality, and yields losses. The virus also induces the information of inclusion bodies, which are the aggregate of the viral protein and host cell components and that can interfere with the normal cellular processes. Molecular dictation and diagnosis. Molecular dictation and diagnosis of the Mpox virus is a complex process that involves the identification of viral RNA or DNA. This is typically done through the use of polymers chain reaction techniques, which amplify small amounts of genetics material to detectable level. Once the virus has been identified, it can be further characterized using the sequencing and polygenetic polygenetics analysis to determine its origin and potential for spread. One of the challenges of the molecular dictation and diagnosis is the high degree of genetic variability within the plum pox virus population. This means that different strains of the virus may require different diagnosis assessed and that the new strains may emerge over time. However, advances in technology and bioinformatics are helping to overcome these challenges and improve our ability to detect and diagnose Mpox virus infection. The next point is chemical control and measures. Chemical control measures are an important tool in the fight against Mpox virus. One approach is to use insecticides to kill the aphids that transmit the virus. However, this method can have negative effect on the beneficial insects and can lead to resistance in the target species. Another approach is to use the antiviral chemical to prevent the virus from replicating within the host plant. This method has shown promise in laboratory study, but more research is needed to determine its efficacy in the field. It is important to note that the chemical control measures should be used in conjunction with other preventative measures such as crop rotation and the use of the virus resistance cultivars. Additional, it is the crucial to follow proper safety protocols when using chemicals to avoid harms to humans and the environment. Chemical signaling and the defense. Chemical signaling and defense is the crucial aspects in the fight against a plumpox virus. Plants has involved intricate mechanism to detect and respond to invading virus, triggering a cascade of chemical reaction that can help them defend themselves. One such mechanism involves the production of specialized proteins that can recognize the viral molecules and trigger an immune response, and other involve the release of volatile chemical that attracts predatory insects which in turn feed on the virus carrying aphids that spread plum pox virus. The next point is development of antiviral agents. Antiviral agents are compounds that can inhibit the replication of the virus by targeting specific viral protein or enzymes. The development of antiviral agent for Mpox virus is a complex, challenging process requiring extensive research and testing. One promising approach to target the RNA-dependent RNA polymer enzymes, which is essential for the viral replication. By the inhibiting this enzyme, researchers hope to prevent the virus from the replicating and spreading. However, developing a effective RDRP inhibitor require a deep understanding of enzyme structure and function as well as its interaction with other viral and circular components. Nanotechnology and delivery system. Nanotechnology hold the great promise in the fight against Plumpox virus. By using the nanoparticles, we can create delivery systems that are highly targeted and efficient. This system can be designed to release antivirus agent at the specific sites, minimizing the risk of the side effects and maximizing effectiveness. One promising approach is to use liquid based nanoparticles. These particles can encapsulate antiviral agents and deliver them directly to the infected cells. They can also be modified to target specific tissues and organs, increasing their effectiveness. Another approach is to use dendrimers, which are highly branched molecules that can be used to deliver drugs and other agents. These molecules can be designed to have the specific properties such as the high solubility or the low toxicity, making them ideal for the drugs delivery. Prevention and control. The most effectiveness way to prevent the spread of Plumpox virus is through the strict sanitation practice. This includes the removing infected trees, burning healthy trees, and properly disposing of all the plants debris. It also important to use the diseases-free planting material and to avoid planting in areas where the virus has been previously detected. In addition to sensation particles practices, there are the several chemical controls measures that can be used to manage the virus. This includes insecticides to control aphids population, which are known transmit the virus, and um, fungicides to protect against the secondary infection. However, it is important to note that this chemical control measure should be used in conjunction with the proper sensation practice and should not be relied upon as the sole method of the prevention and control. Conclusion. In conclusion, the plumpox virus is a serious threat to the agriculture industry. Its symptoms as the devastating and its economic impact is significant. It's crucial that we take immediate action to prevent its spread. We have discussed various methods of the prevention and control, including the chemical control measure and practical advice for the farmer and agriculture worker. However, it is important to remember the prevention is key. By being vigilant and taking proactive measure, we can stop the spread of this destructive virus. And the reference are here. And thank you so much. Allah Hafiz.